this morning we're going to be talking about the wonder of the cell. And I am very excited to give this presentation. This is one I look forward to all of my presentations, but this one is just extra special to me. And you know, as a cell biologist and a molecular biologist, I really like helping people understand the intricate designs of the cell, what goes on in there, all those processes, so that we can stand in even more awe of our Creator God. And I really believe that the designs and the complexities of the cell are one way in which God has made himself known to us. You know, Romans 1:20 says this: "For since the creation of the world, his invisible attributes, his eternal power, and divine nature have been clearly seen, being understood through what has been made, so that they are without excuse." So what God is saying is here is there is enough evidence out there that he has created that people are without excuse. There's no excuse. They can know about the Creator God through what he has created. And so God makes himself known through his creation. Now, it's not perfect anymore. It used to be perfect before the fall, but it isn't. But nonetheless, Romans 1:20 was written well after the fall had occurred, and God says that he can still be known through what he has been made, even though it's not perfect anymore. And we know more about God's creation today than we have ever known before. I mean, think about how much our knowledge has expanded in the sciences since the Bible was written. I mean, we've gained so much knowledge in that area. So I say people are really without excuse, okay? It can't be an excuse. And you know, when I studied the complexities of DNA and the cell when I was in graduate school, it just was hard for me to understand and comprehend how someone could look at all of these things that we were studying and not give God the credit and not believe that God had done this. Instead, it was just the result of random chance over eons of time. And I guess that just goes to show you that the evidence, it's really not about that. It's about our starting points. It's about the interpretation of that evidence in light of our worldview. And we've talked about that a lot, and we're going to continue to do so because that helps us realize that. But I do think that the cell is a perfect example of how God sometimes makes himself known through something very, very small. And I think a passage that illustrates this well is found in 1 Kings. And this is God talking to Elijah here. He says, "Then he said, 'Go out and stand on the mountain before the Lord.' And behold, the Lord passed by, and a great and strong wind tore into the mountains and broke the rocks in pieces before the Lord. But the Lord was not in the wind. And after the wind, an earthquake. And after the earthquake, a fire, but the Lord was not in the fire. And after the fire, a still small voice." You know, Elijah probably expected God to be in all of those other things, and we would have expected the same, but he wasn't. He was the still small voice. And just as God made himself known to Elijah through that still small voice, in the same way, I believe that cells are kind of like the still small voice of God today. And what are they saying? Now, we all know cells can't talk, okay? But what are they saying to us? They're saying God is the creator. God is the designer. I mean, look at all these complexities and all these things that he has done. He's making himself known to us. You know, we live in a very scientific age, and so I think this is one way, again, that we can understand more about God and know more about him. And I want to share with you in this presentation just some of the complexities of the cell. Okay, we can't cover a lot, but just some of those things to help you gain a greater appreciation for God and to visualize just one aspect of the still small voice of God today. You know, it took a long time for us to understand that living things were composed of cells. We kind of take that for granted today, but that wasn't always known. A cell is considered the most basic unit of life. And it wasn't until the late 1600s that the term cell first came about. Robert Hooke was considered the father of microscopy. He developed his very own microscope and was the first one to coin the term cell when he looked at some cork, which is like the outer outer bark of a tree. He looked at that and saw these little individual units, and he called them cells. And a contemporary of his, Leeuwenhoek, who lived also in the late 1600s, he also liked to look at things under a microscope, and he had his own version of that. He's considered, though, the father of microbiology. So he liked to look at bacteria. Now, he didn't know what they were. Okay? He used to actually scrape out the stuff in between your teeth. Lovely. And that from other people as well. And he would look at it under his microscope, and he'd see these little things. And these are pictures that he drew of them. They were moving around. They had different shapes, but he really didn't know what they were. He called them animalcules, little animals. Right? But what they was really looking at was all the bacteria that are currently in our mouth right now. And but even though they could see the bacteria and see the cells, they really didn't know what was going on inside. All they could really see is the outside. They really couldn't see the inside. They used to call them lumps of protoplasm, right? Goo, okay? Lumps of goo. They didn't know all the amazing things that were happening inside. But with the modern microscopes that we have today, we can see a lot of what is going on inside the cells. This is just a picture of one of these types of microscopes that we use. As you can see, it's really developed. This microscope helps you do several different kinds, like phase contrast, fluorescence, things that light up when you look at them. There's different kinds. We've got giant electron microscopes that help us see down almost to the atomic level some of these structures that we have out there. And we'll show you some amazing pictures that we can get today. They're far-fetched from Leeuwenhoek's little animalcules there. This is a hemlock leaf, so we're actually looking at the inside there, seeing some of the chloroplast and other cellular structures. This is a flea. If you have animals, you really love these guys. So that kind of up close and personal. Then we have a butterfly wing made up of lots of different scales. Again, that you don't see when you look at a butterfly, but that's what's actually there. We have these radiolarians. These are small microscopic creatures that are in the ocean, and they have some pretty amazing structural elements to them. This is the cerebral cortex, your brain. This is a fluorescence image, so they've labeled different things inside the cell, and it's fluorescing, and so you can catch a picture of that under a microscope. And then my personal favorite, the dust mite. A lot of people don't like this guy, but he actually serves some pretty important functions. So we can see, I mean, it looks like something out of some sort of alien movie that you would see, but there's lots of these tiny things, all over this room right now. But we can see that. We can analyze those things. We've come a long way in being able to visualize cells and what goes on inside of cells. And a few years ago, there was a computer animation that was produced called The Inner Life of the Cell. That was the name of it, and it was a collaboration between Harvard University and Ex Vivo, which is a scientific animation company. And it was a nice collaboration because the scientists came together with the computer animators to help the computer animators know what these things look like and how they should act in the cell. And I was just amazed when I saw it. I'll be honest with you. I've studied these structures for a lot of years, but I've never seen them put together like this. And I know it's very realistic because, again, like I say, I've studied them. You can go out there in the literature and find pictures of them. But they put them together in an animation so you can actually see how all these things work together. But I want to give you kind of a big picture of what you're going to see. And then we're going to talk about it in more detail because I think the more that we can understand about it, the more in awe we can stand of our Creator God. So the big picture, so here's what's happening. Neutrophil extravasation. What is that? Hey, don't worry, we're going to define these terms because those are some big terms. Neutrophils are white blood cells. Now, we all pretty much know what white blood cells are. They are part of the immune system, and they're very important for fighting against infection and disease. And that's what neutrophils are. They're just a type of white blood cell. Now, they exist in our bloodstream along with the red blood cells. But in order for them to be really helpful to us, they have to leave the bloodstream. We don't want them to stay in the blood. We want them to leave. If we cut our hand and we get bacteria there, we want those white blood cells to leave the blood vessels and go into the tissue to help us fight that infection from the bacteria. So the process of them leaving the bloodstream and entering the tissue is called extravasation. So it's just a fancy word, us scientists. We like to come up with these big, long names for things. But that's all it is. It's just a white blood cell leaving the bloodstream and going into the tissue. What we're going to do now is I'm going to show you clips from the longer video, as well as some clips from this one, and go through what exactly are you seeing? What's going on here? In a way that you can understand. Okay, we're not going to use a lot of jargon and big terms here, but just a way that you can understand, again, to help you appreciate the cell and the creator even more by elaborating on some of these details. So what we want to do now is go back to the big picture. Because as I said, the whole point is to get that white blood cell, that neutrophil, to extravasate, to leave the bloodstream. And there's two parts to that. One is the neutrophil has to bind tightly to what are called the endothelial cells, and those are the cells that line the walls of the blood vessel. So I'm going to show you a picture of that here. So there's the neutrophil, that's that white thing there, bluish white thing. Then we have the endothelial cells. So you're looking at kind of a cross-section through a blood vessel, and the endothelial cells line it, and you have the neutrophil there on the outside. And then you have the bacteria there in the tissue. So you want that neutrophil to leave the tissue at that point where the bacteria are, get in there, and do its job. So that's the first part. So the very first part, though, is the neutrophil has to stop. Okay, it's got to stop where that infection is, bind tightly, and then get into the tissue. So we got to bind first, and then, like I just said, you got to get in. The neutrophil has to get in. It has to change its shape. That was the very last part of the video you saw. You saw it just flatten out like that. It flattens out, and it goes in between the cells and enters the tissue. And that's kind of showing it halfway there. Eventually, will migrate all the way into the tissue and be able to destroy the bacteria. So first, we're going to focus on how the neutrophil bind very tightly to that endothelial cell. That's what most of that video focuses on that you just saw. The very last part talks about it leaving. But so we're going to do that first. So here's another short animation of this. The white blood cells are the bluish white things there. Hopefully it starts. And they're rolling along. They're not rushing like the red blood cells. They're rolling. And what they're going to, the reason for that is because of interactions that occur between proteins on the surface of the white blood cell, or the leukocytes, and the endothelial cells. So these proteins are constantly binding to one another and breaking, binding and breaking, binding and breaking. So that's why it's rolling. Okay? It's not rushing through. The red blood cells, they don't do that. And you want the white blood cells to roll. We don't want them to rush. Okay? We want them to check out what's going on. We want them to stop if there's a problem. There's a very good reason for that. So here's just a snapshot, again, showing different proteins. The endothelial cell, its protein on the outside of the cell is called P-selectin, and then the neutrophil has a protein called PSGL1. And those aren't necessarily important for you to remember, but they're interacting with one another. They're forming and breaking, forming and breaking. Well, okay, that's fine, but we got to get to stop. Okay, we don't want that to continue to happen. We want it to stop where we need it to get in. So how does this white blood cell just rolling along? How does it know where those bacteria are? It doesn't have eyes. It can't see it. Okay. It doesn't know. So how does it figure that out? How does it know, oh, this is where I need to stop, and this is where I need to go in? Well, when you have a bacterial infection or a viral infection, your body, especially when we're talking about bacteria, sends out perfumes, okay, so to speak, attractants to say, come here and help, okay? This is where we need these white blood cells. We need them here. And they come in lots of different forms, chemokines, interleukins, prostaglandins, leukotrienes, all of these that you see here are being released here in the tissue, and they're going to go out to these endothelial cells and become attached to them. And that's going to be like a signal, like a flag going, help, help, okay? You guys need to come in here and help us out and take care of this problem. So that's what's going to happen. So we're going to talk a little bit about how these endothelial cells are reacting and what they're doing to get these neutrophils in here. So the first thing that happens is, and this kind of makes sense, this is a pretty easy thing to do, because those perfumes are coming to those endothelial cells, it causes them to produce more of this P-selectin. Now, what happens when there's more of these here? That's going to slow it down, right? Because it's forming more interactions. As it's rolling, it's going to start going slower as it hits more of that P-selectin. As that's the key to tell that neutrophil, hey, this is an area that's got a problem up in here in the tissue. You need to slow down and check this out, so to speak, and check out what's going on here. Okay, but that's not enough. That just slows it down. We got to actually stop it. We've got to bind it, stop it, and get it in. So another step has to happen. And one of the things that these endothelial cells would do is put something on their surface called a chemokine, and that will interact with the receptor on the surface of the neutrophil. That's what you're seeing right here. So the chemokine on the surface of the endothelial cell binds to the receptor on the surface of the neutrophil. And that's what you see a snapshot of here. So the chemokine, again, it's a perfume that's being made by the tissues. It's being put out on the outside of the cell to say, like a flag, hey, come here. So it attaches to that receptor there. And what you're seeing here, too, this blue and greenish stuff, that's the plasma membrane. That's a membrane that surrounds all our cells. It sort of holds all the goo together, all the stuff that's in there together. You got to have that. So that's what you're seeing there. And so if you would go down in here, this would be in the inside of the neutrophil. So that's what we're going to see next, because what happens when those two things bind is an amazing process now. Is going to be, it's like a signal, okay? It's going to make a change in that neutrophil so that it can bind even more tightly, because this still isn't enough, okay? It's a multi-step process. Now imagine, if you will, though, this takes a matter of seconds. It will take me almost an hour to explain it, and I'm not even giving you all the details. Your body can do this in just a matter of a few seconds. So next we need to go inside the neutrophil to see what happens as a result of this. So that's what the camera is going to do here, basically, is take you inside. So here's the plasma membrane up here. So it's like you're inside looking up. And what we're going to see, it's going to show us the receptor here. It's floating in the membrane. So this is the outside, and this is the inside. And what this is showing you here, what's this big glob that just appeared? Well, it's actually called a G protein. And that G protein is associated with the purple thing you just saw that kind of disappeared there. That's the receptor. So here's a still shot of that. And there's the receptor. This is a chemokine receptor. It's the one that's interacting with the chemokine, the perfume. And it's floating here in the plasma membrane. This is a cross-section of that. So it's like you cut it in half so you could see it. And this is the inside here. Now, it goes through the membrane 7 times. And there's a lot of these. And this diagram illustrates it really well. So I guess these scientists, I don't know, they needed a break from the lab. They needed some exercise. And they thought, hey, let's go out on the lawn and form a 7-transmembrane receptor. This is what people do. That's what, being in the lab for long periods of time, I guess, can do to you. But and they actually illustrated it quite nicely, okay? You've got 7 people here indicating it weaving in and out of the membrane 7 times. This yellow guy in the yellow here, he's the chemokine, okay? So he's bound to this receptor. And then what's going to happen has to do with this person down here. They're not going to illustrate this anymore. And I think they did a pretty good job. That person right here, okay, that part of the receptor actually is going to help interact with this G protein. Okay? And this G protein is going to be really important because it becomes turned on. It's like turning on a switch. And it's like knocking over a domino, which knocks over the next domino, and the next domino, and the next domino, and the next domino, and so on and so forth. All because the receptor, this chemokine bound to the receptor, and it's like it flipped a switch. And these dominoes, which are really proteins, a whole series of proteins are going to be turned on inside the cell, one after the other, after the other, after the other. And why do you need to do this? Because you've got to get the message from the outside of the cell, okay, which is saying, hey, come here, come in here, we need you, to the inside of the cell. You've got to make changes in there. And in order to be able to do this, in order to get those to bind really, really tightly. So what does the G protein turn on? I said that turns on a protein, which turns on another protein. Well, it turns on a lot of things. Now, don't get freaked out, okay? We're not going to go through all of this in detail, but this is just to show you the amazing complexity of what's going on here. We have a chemokine. This is the receptor here that I just showed you, the one that goes through the membrane 7 times. You've got the chemokine bound to it. Then you've got the G protein right here on the inside of the cell. Now, that G protein, when it gets turned on, it turns on this, which turns on this, which turns on this, which turns on this, which turns on this. Okay, this is the short version here. And eventually, you're going to get a message here to the nucleus. That is the key. You must get a message to the nucleus. Why? Because that's where the DNA is. And that's where the information is to make the proteins that are going to change that neutrophil so it can bind to that endothelial cell. It's like where the key is, okay? Where the little secrets are. You got to get those out of there. You got to get that out of there so something can happen and a change can occur. You want, in this case, what we're wanting to happen is to make specific, to basically release this information. We'll talk about how that's done here in a minute, so that proteins can be made that can be put on the outside of the cell here, on the surface of the cell, and interact with that endothelial cell to get it to bind really, really tightly. So now we're going to travel, okay, all the way from here, all the way down here to the nucleus, okay, to that nuclear membrane. The nucleus has a membrane too, to hold the DNA in, okay, to keep it safe and protect it there. So we're going to go to the very surface of it. We're not going to go inside the nucleus, but we're going to look at the surface of the nucleus. So that's what you see here, hopefully. And there's little pores or holes in the nucleus, and then something comes out. Right? It looks like string almost. And so let's talk about what that is. That is actually something called messenger RNA. Now, DNA is down here in the nucleus, okay, because the nucleus is down there. And that's where that's where the DNA is. Now, as I said, all these proteins have been activated, and they've gotten a message to the nucleus. They've gotten a message to DNA. Say, hey, DNA, these are the proteins you need to. These are the proteins that need to be made. So make a copy of yourself, okay? Make a copy of these particular genes, because genes are what encode the protein. Make a copy of these genes and send it out. We got to get these things made. It's very much like a factory, you know? You're passing the information along, and things are being made as a result of that. So it says, okay, well, we've got to make these particular, this has the information to make those proteins on it. It's a copy. DNA stays where it's at, okay? You don't want it to leave. You want to keep it safe in there. There's a lot of dangerous things outside of the nucleus that could damage it. So you keep it there, and you just make a Xerox, more or less, and send it out. And so that's called messenger RNA because it has the message inside of it. Now, before we go on to actually seeing, okay, how does this messenger RNA or this mRNA made into a protein, because it won't do us any good in this case as RNA. We've got to make it into a protein so we can change, again, that interaction. I want to talk a little bit about the pores that are here, okay, these holes in the nuclear membrane. They're more than just a hole. They're the nuclear pore complex. They are an extremely complex structure. This isn't just some little, like, hole that you would punch in a membrane or in a balloon. They're extremely complex structures. They have a basket-like look, and they're made up of many, many, many different proteins, which is what it's showing here. They're very picky about what goes into the nucleus and what comes out of the nucleus, because you want to keep the DNA safe, and you only want the RNA to leave. You don't want DNA to leave. So they're very picky about that. So very, very complex structures there. So next, we're going to watch what happens to this RNA, because it has to be made into a protein. And so what's going to happen here, these green things are going to come into the picture, and this is the ribosome, or part of it. And here comes the other part. And they are going to read the message. Okay? They're going to make it into this yellow thing, which is the protein. They literally read it, and they do this very, very quickly. And so that's what you see here. So you have the mRNA, and you have the ribosome attached to it. Now, the ribosome is a machine. It reads the RNA in 3-letter words. That's all it reads. It can only read 3-letter words. And each of those 3-letter words stands for one amino acid. And amino acids are put together and put together. As that message is read, they keep growing and growing and growing, and eventually you get a protein out of that. That's what that little yellow thing was that floated away. Now, the ribosome, in and of itself, is a very complex machine. In fact, in human beings, it's made up of 82 different proteins. You got to have 82 different proteins to be able to take an RNA and make another protein. And that's the complexity here. It's a very large conglomerate of proteins, and you see the messenger RNA in here, and it reads it, and this is the amino acids. Again, reading it in 3-letter words, and each of those stands for an amino acid, a different color segment you see here that's growing and growing and growing, and eventually that becomes the protein. Now, once the protein is made, it has to go somewhere. So how does it know where to go? Because again, it's not just floating around. Don't have that idea of the inside of the cell. It's not just a bunch of goo, okay, with things floating around. It's very highly organized. So it has to go somewhere. And in this case, this particular protein that's made here, this blue one, attaches to a transporter protein, and it's transported to another organelle called the mitochondria. And I talked about that a little bit yesterday in my talk. Those are really the powerhouses of the cell where we get our energy made from. And so this just shows you a snapshot of that. Now, how does that transporter protein know that it should bind to this protein and that it should go to the mitochondria? I mean, how does it know that? Well, the protein, this blue thing right here, has specific sequences in it. Right? And so those amino acids come off. There's a specific sequence, and that specific sequence says, one of those says, you must bind to the mitochondrial transport protein. And then you must go to the mitochondria. And it's even more complex than that, because once it gets to the mitochondria, get this, it has other sequences that tell it exactly where to go once it gets inside. It's very, very specific. It's just like, when you write a letter to someone, you put on there the street number, the street name, the city, the state, and the zip code, right? So make sure it gets to the right place. Well, that's exactly like these proteins, what they have in them. Not only do they have a function, they do something in the mitochondria in this case, they've got a street number, a name, a city, a state, and a zip, okay? All in their sequence to get them to the right place, not only to get them to the mitochondria, but get them to the right place in the mitochondria. And if even one of those is messed up, people can have very severe disorders as a result of that. I mean, one little thing is wrong, and all of that can be, lead to a lot of problems in the individual. Now, sometimes proteins have to undergo further modifications. They can't just, bind to the transport protein and go where they need to go. A lot of times, proteins need to have sugars added to them or other changes that have to occur. So in that case, we have to take them to the ER. Now, this is not the emergency room, okay? This is the endoplasmic reticulum, okay? We'll just call it ER. That's a little bit easier to say. So we're going to take them to the ER. So that those changes can be made. Now that's what you're going to see here. This particular RNA is encoded for a protein that needs to go to the ER. So there's a transport protein that brings it here and sets it down on the ER, a pore or a hole in it, and it still reads the RNA, but you notice the protein here is going inside the ER. It makes it right inside so that the modifications can occur. You see a snapshot of that here. Again, we didn't see the transport protein, and something the video really didn't show, but a little bit of that protein is made, just a little bit, when that ribosome binds, and that's enough. That's a signal that says, bind to this ER transport protein, take it to the ER. So it knows to do that, just like for the mitochondria. So it takes it to the ER, and again, the ribosome reads the messenger RNA, and the protein is made, but this time inside the ER, not just out there in the cell. And so, again, the whole point of this, and this is going to be important for the proteins that we're dealing with. We need protein to make our change occur. Remember, I'm going to keep taking you back here. The whole idea of this, and that's why I say it's easy to get lost in the details, isn't it? I mean, why is all this happening again? Because we want that neutrophil to bind really, really tightly to that endothelial cell. So this is what it takes to get it there, okay? We've got to make changes. We've got to put certain other proteins on the surface to get those to bind really, really tightly. So, it takes us a while to explain that, but that's what's going on. Now that the proteins have been modified inside this ER, okay, once they've had their changes occur, they next need to get to their final location. This is my favorite part of the entire video. And so I'm going to show you two clips because I like it so much that illustrate this. And maybe you saw me smiling extra big when we were watching it. So here we go. So these little vesicles kind of pinch off the ER that have the proteins in them, and then you're going to see this guy right here. Okay? This is called a motor protein. And it's got a very big cargo here from the ER that has those proteins inside of it, and it's walking. It's walking along structures inside the cell. Let's see another photo of that. Here we go. Okay, this is called a microtubule, and this motor protein literally walks step by step by step. Every time it takes a step, it uses energy. That's why you need to eat your Wheaties in the morning, okay? This is going on inside of every cell that you have in your body all the time, okay? All the time. This isn't just specific to the neutrophil. This is happening in all of our cells. That's what's, and a lot of these processes that I'm talking about isn't exclusive to the neutrophil. So I showed my daughter this. My daughter's 5.5, and I was trying to get her really amazed. And she says, "Is that going on inside your body too, Mommy?" I don't know. I may have freaked her out a little bit more, but I said, yes. And so she just kind of stood there and watched it. Because it is so neat and so amazing to think that these things are walking inside our cells right now. And so here's just showing you the vesicles themselves, the little pieces of membrane that have the proteins inside of them, because they have to get to their final locations. And I want you to remember, too, this is not just some fancy computer animation. This is reality, okay? We know that this is what happens. In fact, I just read a paper literally last night, or a news story about that they are able to visualize this even better now inside the cell, how these things walk. So here's another picture showing it. This is the motor protein itself. You have many different motor proteins inside your cells. Some walk this way, some walk this way, some walk both ways. It depends on where they need to go. They have different tails here. This is called the tail of the protein. And the vesicle itself has certain proteins, which you can kind of see here on the surface. So depending on where the proteins inside need to go, the proteins on the surface differ, and they associate with different motor proteins. Very organized to get it to the right place at the right time. Now, they're walking on a structure called microtubules, and these are the highways of the cell. That's the best way I know to describe it. This stuff is not just floating around inside of there, okay? It's taking the highway. It's being carried here by this motor protein. It's called the head of the protein, and it walks step by step by step, which I'll kind of show you here, down this microtubule. Just like we would walk. When I see this, it reminds me of the videos, or pictures you see of women with those really huge things on their heads, and they walk just effortlessly, down the road, and you think, wow, how can they do that? Well, that's exactly kind of what's happening here. They're carrying these huge, huge cargoes, which are much larger than they are, down these microtubules to the place that they need to go. So where are the proteins that we're interested in inside of here, where are they going? Well, the ones that we're interested in are going to go to what we call the plasma membrane. Remember I said that's the membrane that surrounds the entire cell. And so they're going to go to that membrane. Because again, the whole point is to get a protein out there on the surface, stick it out there, it's going to make them bind really tightly to one another, that neutrophil and that endothelial cell. So here we're going to see the release of the proteins from the vesicle, boom, outside to the outside of the cell. Now, some of those proteins leave, and some of them stay in the membrane, which you can see there, and I'll show you some still shots of that. The vesicle, and this is kind of hard for me to explain, but the vesicle that was walking, okay, that was attached and walking, it's come to the membrane and it fuses with it. It kind of melts, okay, into it. And as it melts into it, the proteins that are inside get released. And the blue here are secreted proteins. They go somewhere else and do something else. We're not really interested in those right now. What we're interested in is these guys, these yellow ones. They stay in the membrane. They don't leave. They become part of that neutrophil membrane. The change that needs to occur to allow them to bind tightly to that endothelial cell. Now, again, all of these changes that have occurred, all that we just talked about, what did it do? It produced this protein that got on the membrane of the cell. And I'm just giving you the basic version. It's actually much, much more complex than this as to what happens. But this is the idea. This is the gist of it, to get that protein onto the membrane of the cell. So we're going to do a little bit of review in this next video, because that's always helpful. Again, remember, the chemokine on the endothelial cell bound to the receptor on the neutrophil, and then we went inside the cell to see what happened. It turned on the receptor, which turned on the G protein, which got a message to the nucleus, and sent that out, made the proteins that then are released from the cell, but the ones we're interested in actually stay in the plasma membrane of the neutrophil, and that particular protein is called integrin, and that we're interested in, and they kind of cluster together all in one place. That's what you're seeing there, sort of come together. And so here's a snapshot of that. Again, this stuff up here, you might be thinking, what is this? It looks like it has an atmosphere to the cell. This is called the extracellular matrix. Think of it as the glue that kind of holds all the cells together. Okay, it's the stuff kind of in between the cells. And this is the plasma membrane of the neutrophil, and then you have the integrin there. Now, in this next video, what you're going to see is that integrin has to change shape. And it changes shape by basically extending itself. Boom. It extends itself, and this is the endothelial cell up here, and this is a protein called ICAM that's associated with the endothelial cell, and that is the final step, at least as far as the binding occurs. You need that integrin to bind to that ICAM there, and that's what you see happening. So all of that we just talked about, the whole point was to get this integrin. It changes shape, extends itself, binds to the ICAM, and finally, we've accomplished the ability to stop that neutrophil dead in its tracks, okay? We've got to stop it, because that's where we want it to enter the tissue. So that's what you see represented here. We've got the endothelial cells up here, and we've got the ICAM here, and we've got the integrin here. So they've had that final interaction, that stopping interaction. Now, what happens next? Because remember what we said, there's 2 steps. You got to bind, it's got to bind, and it's got to change shape to get in. So how does it change shape? Now, how does it know to change shape? Because that's the next thing that makes sense that it would have to do. Well, now, we're not going to go into all the detail on this one, because the video doesn't either. But when this ICAM binds to integrin, guess what? You set off a whole chain reaction inside the cell all over again. This integrin turns on a protein, which turns on another protein, which turns on another protein, just like we saw before, only this is a whole different set of proteins, okay? Because these are proteins that are not wanting it to bind, but rather to flatten out, to change shape, and get into the tissue. Whole different set. And scientists are still trying to figure out how cells keep all this straight. It's all in there. How does it keep it straight? They've even believed there's certain proteins called scaffolding proteins where they'll fit in the order they're supposed to be activated in. So they all just fit there waiting for that first one to be turned on, and boom, boom, boom. Because how do you organize all of this? It's just so amazing. So the idea here now would be to get a message to the nucleus to say, hey, we need to change shape. So now you need to produce proteins that are going to cause a shape change. So to flatten that cell out so it can get into the tissue and do this. So a very, very complex thing that's going on here. Totally different set of reactions, totally different proteins, totally different DNA that's being told to do something, totally different RNA. All of this in a very short, like literally seconds, period of time. Just amazing. So next, again, like I said, the neutrophil has to change shape. And we've done the binding, so we did that first part. We said that's the first thing that has to happen. And then we said it needs to get in. It needs to actually kind of really morph here, change shape, and get in between these cells and get into where the bacteria is. So I'm going to show you a video of that here again. There's the white blood cell rolling along. So all those interactions that took me forever to talk about just happened like that. And it changes shape. It literally just flattens itself out, so it becomes very two-dimensional. And then it will squeeze in between these 2 endothelial cells and get into your tissue. Now, what happens is a massive reorganization of the skeleton of the cell. Not only do you have a bony skeleton, but the cell itself has a skeleton. So this is showing you the inside of the cell here. I'm going to show you the skeleton. A lot of these proteins are considered part of the skeleton. This is actin. Actin is a very important part of the skeleton of the cell. It's the purple stuff that you see here. The green is other proteins that link those together. Now, we're not going to talk. The video goes into a little more detail, but we don't have time to talk about all of the skeletal elements here, but these are some of them. These are called spectrin here. They kind of look like spokes on a wheel, is how they describe it usually. And then you see the actin down here. It lies just beneath the surface of the plasma membrane. And it's composed, like I said, of a lot of different protein. It provides shape and stability to the cell. It's rigid yet flexible, much like our bony skeleton. I know for as many times as I've seen my daughter fall, I'm glad she's got a somewhat flexible skeleton, okay? If it didn't have a little bit of give, right, it would shatter and break. So it needs that flexibility. But some cells, okay, unlike what our bony skeleton can do, the cytoskeleton can actually massively change so that it's very, very dynamic. We're not that dynamic. We can't flatten ourselves out and go under doors unless we're like Mr. Incredible or something. It's not going to happen, okay? That's not possible. We can't do that, but our cells can. They can actually morph, so to speak. They can flatten out. They can change shape and do that. And one of the ways they do that is by changing the actin, which again you see here. If you go just beneath the surface of the cell, the actin is like steel girders, okay? It's very parallel rows that are bound together with other proteins. And again, it depends on where you're at in the cell, actually what the actin looks like, because actin is a major component of that skeleton of the cell. This is kind of showing you that. This is a cross-section of, went just beneath the plasma membrane, just beneath it, showing you the different ways the actin can look depending on where you're at in the cell. really near the cell membrane or the plasma membrane, it's very tight. You can't see individual filaments, real tight together, like steel girders on a building. But as you go further down into the cell, it becomes like a spider web, kind of meshy and networky. Again, the idea is to have rigidity yet flexibility. It's got to have both. It has to have kind of an even amount there. Now, when you get down into that meshy part, there's a lot of activity that's going on. These actin filaments are constantly forming. This is a process we call polymerization, okay? But we can just say forming. Forming, they come together, and unforming. They'll even depolymerize. Parts will fall off, and parts will form and fall off and form, depending on what the cell needs to do and when it needs to do it. And again, how does it know to do this? Because of that message that was sent to the DNA that's now produced proteins that says, hey, actin, you need to do this. This is how you need to change. We got to flatten out. We got to get in there. And so that's what's going on there. So this just shows you another close-up of the actin. And there's lots of other proteins that are associated with them. This is an actin binding protein here. And then these are little individual actin units that come together, like I said, to form the actin. It's very actively doing this. Now, the same is going to be true for microtubules in the cell. Now, we talked about microtubules earlier. They're the highways of the cell, okay, where that little motor protein walks down. They're an active part of the cytoskeleton, too. And we're going to see here kind of their activity. Now, when the units come together here, they actually form tubes. They're not filaments like actin is. They're actual tubes. And they polymerize as well. They come together or form. And then they can also depolymerize, which means they lose units. They come apart. They sort of look like something that's frayed, you know, like material that's fraying. That's what they actually call it fraying that happens here. So it's falling apart. So that's constantly happening, too, to change the shape of this cell. That's the whole point. Remember, keep that in mind. We've got to make things different. So they're forming and then unforming. So now I'm going to show you some other still shots here of what happens. Okay, here's the endothelial cell. So it wants to squeeze between these two cells right here. So it forms what's called a leading edge. So again, that integrin-ICAM interaction occurred. It got a message to the nucleus to say, hey, actin, you need to change, okay? You need to start growing and pushing out. It's kind of like if you were on the inside of a balloon and you were pushing out on one part of it. That's what's happening here. That actin is building, 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 and then it builds, it pushes out and forms what's called a lamellipodium, which is a fancy name for foot. Foot that forms here, and it pushes out and to start flattening out the cell. And that continues to happen on this end, and on the trailing edge, something is actively happening too. The microtubules are starting to break apart. They're starting to fray and get smaller so that the trailing edge can kind of catch up with that leading edge. So again, getting that, again, you saw that really well in the video, and I'll show that to you again, where it just flattens out. So you have a leading edge, and then the trailing edge is kind of trying to catch up to it there, and eventually, yay, we're inside the tissue, finally. As we go between these two endothelial cells, it will regain its three-dimensional structure. Again, there'll be other signals that tell it to, hey, you know, that's over with. Now we need to go back to the way we looked before. So that's the process of extravasation, of getting the white blood cell into the tissue. So again, here it goes, boom. Okay, so this is the leading edge here, and this is the trailing edge. So, and you'll see that better here. That's the foot-like structure that's formed, and the leading edge catches up, and it's just flat as a pancake. And it flits in between these two endothelial cells, and we're finally done. Right? We finally got all the way from, okay, this is what needs to happen, that message outside the cell to inside of the cell to back outside the cell again to make these changes occur so that our bodies can be kept safe from foreign invaders like bacteria and viruses. You know, again, Romans 1:20 says that we can know about God through what he has made. And in today's world, we can clearly see the complexities of the cell and thus know of God's intelligence and power. Cells are very much the modern version of the still small voice of God. But you know what? There are people out there who still do not believe that. And it's amazing to us. You know, we sit here and we look at this and we talk about this, and especially as myself, someone who's really studied it in detail, and you think, how can they? I mean, fine, someone can look at a sunrise and sunset and say there isn't a God, but how can you look at this and say that there isn't a God? And what we learn from this is that while evidence, for example, the complexity of the cell, while it's a good argument for God, the existence of God and the truth of his word, it's really not the best argument, because evidence is always going to be subject to the rescuing devices of the evolutionists. There's always going to be an Oort cloud that sends comments. That explains, what they need to explain. There's always going to be, for them, some mechanism or process that caused this complex structure or mechanism in the cell to come about that, maybe doesn't work anymore today, or, it only, we just haven't found it yet. Okay, and then you're kind of at a dead end, right? And, we think about that we have problems with this, but you know what? Even Jesus Christ had problems with this, that people didn't believe the evidence. Matthew 28, 16 and 17, and the 11 disciples went away into Galilee to the mountain which Jesus had appointed for them. When they saw him, the evidence, they worshiped him, but some doubted. They had God incarnate standing in front of them, and they refused to believe. I mean, that's just amazing to us. So you know what? But what's really neat, and then we see throughout Scripture, but especially here at the time of the resurrection, in Luke 24, we see several examples of Jesus reasoning with the disciples from the Scripture. And what happened? They believed. It wasn't enough for them to just see Jesus Christ. He had to reason with them from the Scriptures, just like we need to do. And beginning at Moses and all the prophets, he expounded to them in all the Scriptures the things concerning himself. So they rose up at that very hour and returned to Jerusalem and found the 11 and those who were with them gathered together, saying, "The Lord is risen indeed and has appeared to Simon." They believed. He reasoned with them from the Scripture, and they believed. And while I just stand amazed when I look at the complexity of the cell, and I, again, just want to say, my God, how great thou art. And it is a good argument. There's no doubt about it. Evidence is a good argument. And Dr. Lyle, in his next presentation on nuclear strength apologetics, is going to tell you how to use evidence in the creation apologetic. It's very, very important. But at the same time, I realize the ultimate argument is to contrast our worldview with theirs and show them that all knowledge and truth is based on the Word of God. That's the most important thing.